Hi there. I'm Bob O'Neill, and I'm here to give you the keynote talk. I'm sorry I can't be physically there, uh, but due to circumstances some of you probably are aware of, I am not. Weedy has a little bio on me that she will probably mention, but I am retired. I live in Delaware. But I spent most of my career at the Food and Drug Administration. So I thank you for the invitation to give this particular talk. And with that, let's get into it. I would assume that you will have access to these slides. So I will not go into detail on some of the slides because they have a fair amount of material in them. So you will have them available to you to look at your discretion. So with that, I will start the talk. Okay, so the title of my talk is Regulatory Science, Statistics, Innovation, and Education Models. The disclaimer here is while I consider and discuss issues of regulatory importance, you need to recognize that I am retired from FDA and have no official relationship with FDA, and these are my own thoughts and positions. So this is the outline of the talk topics. I will provide you a motivation for this talk. I will then give a brief discussion of what I think is regulatory science, what it is, what it contains, something about evidence generation to meet regulatory standards. This is something which may not be well known, but it may be well known to you all because this is the business you're in the pharmaceutical industry. Then I will spend some time on the current environment for innovation expressed in regulatory guidances, and with a brief discussion of the impact of the COVID pandemic. And then biostatistics, which in my mind is about clinical trials, methods, applications, course content, and use of real-world examples in the pharmaceutical development space, and how this relates to the current education and training modules. I'll talk about two of my personal experiences, one with giving a course at the Harvard School of Public Health for 4 years, and another where I gave a year-long course in the European course in pharmaceutical medicine in Basel, Switzerland in 1989 to 1990. And then I will spend some time on the need for academic training in regulatory science with some concluding remarks. So the motivation for this talk is my personal feeling that the statistical framework underlying drug development and drug regulation is increasingly more complex and fundamental to providing critical evidence for product approval. Also for efficient trial and program planning, monitoring, safety assessment, data access, and integration of all of these for decision-making. The regulatory term substantial evidence of efficacy and safety relies on randomized controlled trials and the statistical principles underlining their design, analysis, and interpretation. I will be covering many regulatory guidances that relate to this particular aspect. In my mind, regulatory science is a mature body of content, continuously evolving, yet not taught in the university or medical school setting. And many in the industry are ill-informed about the key aspects that would mitigate failures in development had they been exposed to these concepts in regulatory science. So with innovation being the modern mantra for pharmaceutical development and science, there needs to be new ways of translating regulatory science into training, education models, and team collaboration in the pharmaceutical development and regulatory space. This essentially is a culture change. So how and when did this change in culture begin? Actually, it began in 2004 with FDA's Critical Path Initiative. That was FDA's national strategy for transforming the way FDA regulatory medical products are developed, evaluated, and manufactured. The initiative was launched in March of 2004 with the release of FDA's landmark report, Innovation, Stagnation, Challenge, and Opportunity on the Critical Path to New Medical Products. That publication diagnosed the reasons for the widening gap between scientific discoveries that have unlocked the potential to prevent and cure some of today's biggest killers, such as diabetes, cancer, and Alzheimer's disease, and their translation into innovative medical treatments. Sounding the alarm on the increasing difficulty and unpredictability of medical product development, the report concluded that collective action was needed to modernize scientific and technical tools, as well as harness information technology to evaluate and predict the safety and effectiveness and manufacturability of medical products. It was everybody's problem. So here's just a screenshot of that article. And there was another article by Janet Woodcock, who is one of the authors of this, and Ray Wolseley. I won't spend any more time on this. So FDA's Critical Path Initiative followed up with another report, which requested input from external sources, including the industry, other people who wanted to weigh in on this. And this initiative triggered many discussions among stakeholders. And the enumeration of the leading areas for scientific improvement in the development process were what was essentially came out of that effort. And some of the key areas that were highlighted were the development and utilization of biomarkers, modernized clinical trial methodologies and procedures, enhance the use of bioinformatics, including disease modeling and trial simulation, improvement in manufacturing technologies, and consortia, development of consortia involving clinical path activities. So you can see that was 10 years ago, and well over 10 years ago. And that stimulated what we're facing today. So that leads us to the current environment of innovation. There are many aspects to innovation, many areas of application, but a few that I might focus on is quantitative safety analysis and surveillance. This is critical for a lot of the safety problems that have evolved over the last 10 to 15 years. And in particular, many of the clinical trial study design advancements, enrichment designs, multi-regional clinical trials under the ICH umbrella, adaptive designs, seamless phase 2-3 designs, basket trials, and master protocols. These are all new innovative strategies in the clinical trial space. And then, of course, many different ways of data collection that didn't exist 10 to 15 years ago. Mobile devices, electronic data capture, software as a medical device, real-world data, the Sentinel system. And then finally, a change in the way clinical trials are going to be monitored, not necessarily people at the site all the time, but real-time central monitoring versus site inspection, the updated good clinical practice guidance, quality by design, which is in the new ICH E6 updated guidances. Many statistical methodological changes, ICH E9 update, which essentially has the whole area of missing data and S-demand and sensitivity analyses, biomarker qualification, enrichment designs, and biomarker development and qualification. These are just the areas of innovation that you've probably heard about. So here's my definition of regulatory science. I think it includes all the regulations that establish the scientific criteria for the efficacy and the safety of medical products. It also includes all of the guidances, which I will get into in detail, issued by FDA, EMA, the European Medicines Agency, and the ICH, the International Conference on Harmonization, that contains substantial scientific content and advice. It's the data that is now out in the public domain from experimental clinical studies that are used for modeling, meta-analyses, simulations of clinical trials. It's the science of regulatory statistical evaluation of evidence, which is practiced by the FDA. And there's a huge infrastructure that is behind that evaluation of evidence. It, for the most part, it's not achievable with any literature peer review process. So the refereeing process is not a substitute at all for the science of regulatory evaluation, in particular, the intensity of the statistical evaluation. And then finally, you have some case studies that are all publicly available. These are FDA reviews of sponsors' clinical trials and their program of evidence generation, the advisory committee meetings that are held in the public domain and are videotaped, and that serve as lessons learned for some decision-making. The methodology development, new tools to evaluate evidence. One example being a qualification of a statistical method called MCP mod, which essentially handles modeling of dose response at the same time that it adjusts for multiplicity control. And the stimulus for innovation constantly evolving with the science. So FDA has a definition that they put out. It's the science of developing new tools to assess the safety, efficacy, quality, and performance of all FDA regulated products. The knowledge gained from the science and research increases the certainty and consistency of regulatory decisions. This is the report that was issued in the year 2011. So it's been around a good 10 years. And it describes what regulatory science is. I personally think that regulatory science includes more than this particular report includes. I gave you earlier, several slides earlier, my definition of how much it includes, including case studies and advisory committee meetings and whatnot. So the regulatory science framework, if you will, is the science of developing new tools. This is FDA's definition. We'll discuss the role of many science-based published guidelines, which I will spend a considerable amount of time on. And these guidelines are issued by FDA to implement the regulations and provide sponsors strategies to plan study designs, good clinical practice to conduct and monitor studies, appropriate patient safety assessments, and data collection. These are essentially the roadmaps for what the industry should follow to develop and generate their evidence, to analyze it appropriately, store it, be ready for auditing and whatnot. So with all this background and the importance of regulatory science, My main position in this talk is that there has not been an effort in training in this field in the academic space, and I think something needs to change. As a result, scientists working in this field, whether as a regulator, an academic, or in the pharmaceutical industry, have to learn it on the job, a very inefficient way to learn. So a core component of regulatory science, in my mind, are the case studies of FDA Advisory Committees that evaluate substantial evidence of efficacy and safety, risk-benefit assessments, and study design discussions. If the advisory committee is held on the White Oak campus, they are webcast, archived, and available under an Adobe Connect format. More recent meetings during the pandemic have been restricted to non-in-house person-to-person meetings, and many of them are Zoomed and are on YouTube, particularly the COVID-19 vaccine meetings that have been held over the last 6 or 7 months that included discussions of the Pfizer, Moderna, and the J&J vaccines and the evidence supporting them. In addition to that, there's many FDA slide presentations and industry presentations and briefing packages that are submitted by the industry to FDA, along with the minutes of the meeting and the voting results, if held, as well as the video. So this is a lot of material. This is publicly available to include in training and educational components. I will touch on some examples that I used in a course that I taught at the Harvard School of Public Health and described in a published article in Chance magazine. The other core component of regulatory science are the guidances. These are extensive regulatory guidances on multiple topics of relevance to clinical trials, disease-specific study approaches, good clinical practice, quality assurance, and safety assessment. It's not only FDA's guidances, but their European guidances, Japanese regulatory guidances, and the International Conference for Harmonization guidances, which are consensus guidances for all international regulators. So most statisticians are probably aware of these recently issued regulatory guidances that have substantial statistical content. They, for example, adjusting for covariates in randomized clinical trials, adaptive design clinical trials for drugs and biologics, multiple endpoints in clinical trials, an updated E9 statistical principles for clinical trials that included estimands and sensitivity analysis in clinical trials, meta-analysis of randomized controlled clinical trials to evaluate the safety of human drugs, and E17 general principles. E17 is a ICH guidance, general principles for planning and design of multi-regional clinical trials. As you can see, these have all been issued within the last 2 or 3 years, and more are coming. This is a second slide in cataloging recently issued guidances. E16, another ICH document on biomarkers related to drug or biotechnology products, qualification process for drug development tools, and there was a statistical approach known as MCP mod that was qualified by the European Medicines Agency and FDA within the past several years that combines dose response estimation with multiple comparison control, a real, if you will, success story for statistical methodology tools being approved by regulators. And of course, more recently, the COVID-19 master protocols evaluating drugs. This is, as we speak, the way many of the multiple treatments for COVID-19, not necessarily the vaccines, but treatments for COVID-19 are being evaluated in umbrella trials and basket trials. This is an example. I won't spend much time on it, but this just shows you the qualification opinion of that statistical procedure, MCP mod, by the European Medicine Agency on the left-hand side, and the Office of Clinical Pharmacology and the Statistical Office also qualifying the MCP mod approach. So following up with more guidances that have substantial statistical content, there is a guidance that speaks to interacting with FDA on complex, innovative trial designs for drugs and biologics. That was issued last year. There was an E6 good clinical practice update, which is integrated to the addendum to E6, came out in March of 2018. That gets into the quality by design issue, central monitoring. There's an FDA guidance on the conduct of clinical trials of medical products during the COVID-19. So it's essentially to put some goalposts out there for the industry to follow in designing trials to deal with the COVID pandemic. There's also a statistical only guidance on statistical considerations for clinical trials during the COVID-19 public health emergency. So you can see these are all guidances that have been issued within the last year and a half to 2 years. There is another important guidance on evidence flexibility, and this has updated a guidance that was probably 15 years old, and it's to accommodate the new examples for the quantity and quality of evidence required for rare diseases, where it's hard to find patients. It may be hard to have more than one study. So it speaks to the replication of evidence. It speaks to the quality of evidence, and where there are areas of flexibility to accommodate the study of rare diseases, serious diseases with no currently available cure. And there are 4 FDA guidances on this particular topic. Here they are. I'm not going to spend much time on them, but the web address for access to these guidances that are on FDA's website are provided on this slide. And it, in particular, spends some time, one of the guidances spends some time on natural history studies for drug development and other principles for co-development when you have a diagnostic that has to be used along with a drug, a therapeutic. So they have to be co-developed, as it were. And there are webinars and slides on these particular topics that are also available on FDA's website. In addition to this, there is a whole other area of electronic source data for clinical trials. And the fact that if the clinical trial is being submitted to the regulatory agency, there needs to be a format for the electronic data to follow, including the data standards for the clinical trials. And there are FDA guidances on these topics that are constantly being updated. And here are 4 guidances with the web address for how one accesses these particular documents. So I've given you so many guidances that are probably overwhelming in terms of the number and breadth and what they cover. But this particular schematic is intended to provide you with a sense of where in the whole development process do these guidances come into play, because they're important for the whole drug development team to be aware of how the different guidances interrelate with each other. So these lines and arrows show where different guidances come into play in phase 1, phase 2, and phase 3, and in post-market, post-approval areas. So these are not extensive. It's not all the guidances, but it shows you how many of them are involved in different dimensions of the development of phase 1, phase 2, and phase 3 in a drug development program. And I haven't even dove into some of the finer points of adaptive designs and seamless phase 2, phase 3 type designs. So as the development becomes more complex, and these guidances are issued to deal with the complexity, the academic world is unaware. At least that's my position. I am unaware of any of this work being taught in the academic space. So the scientists and clinicians that enter the pharmaceutical industry and the regulatory enterprise mostly come from the academic community. Most in the academic community are unaware of basic regulatory standards of evidence, how it is developed, evaluated, monitored, audited, and the rigor required of the team to plan, conduct, analyze, and interpret experimental evidence. In my mind, it is a learn-on-the-job exercise, increasingly inefficient. So my position is, how do we shrink the learning curve? Is it time to incorporate more regulatory science content in the curricula, including biostatistics department? And what content might that consist of? So one area is real current examples of evidence generation and evaluation based upon clinical study designs, observational studies, meta-analyses, external data sources requiring modeling, propensity score matching, causal analysis thinking, missing data and estimand sensitivity approaches. These are all incorporated in real, actual examples that FDA faces and sometimes are brought to the advisory committees. Regulatory guidances, many of them I just went over in somewhat detail. Case studies in the public domain. These are all on FDA's website, as discussed earlier. And the documentation of regulatory approval decisions. If a drug is approved, the documentation of that approval decision and the statistical review, the clinical review, the pharmacometric review, these are all in FDA's databases, and they're available at Drugs at FDA. So why raise this topic now? In my mind, it's about time and well past time. The statistical issues involved in the pharmaceutical development and fundamental to success of the efficiency of programs to bring safe and effective therapies to patients. I mean, that's the critical point. Statistical issues are really the fundamental underpinnings of much of what we now do. Most statisticians are not exposed to these issues in their training currently. In my mind, I could be persuaded otherwise. Also, most clinicians who come from the academic program, who become academic investigators, or are maybe supported by the NIH clinical grants, and then are involved in IND studies, are unaware of most of the core regulatory criteria and rigor required. they're mostly interested in and trained for a different role, and that is to publish. So considerable concern for these inefficiencies in drug development and the lack of success and failures that might be avoided or minimized if better informed are the reasons why it's about time to change the culture of the academic teaching of regulatory science. And this particular commentary is not just my view, but it's about the view of this particular issue that is coming from a group that has been devoted to advancing a vision for regulatory science training in the academic world. These people are in the academic world, and they've recognized this need. So this particular paper recounts a workshop that was held on regulatory science core competencies and curricular guidelines. And I will leave it to you to read this particular, it's a good read. Here is a brief summary of what I have done in my professional life to try to make this particular topic come alive. In 1989, FDA supported a program that I put together at the University of Basel in Basel, Switzerland. And I moved for 1 year to Basel. And I had observed that the European regulatory efforts and guidelines for clinical trials and drug development really was absent any statistical input, and actually did not realize the level and depth of rigor that statistics brought to the problem. At that time, we're talking 1989, there was no regulatory statistical expertise in the United Kingdom, in France, Germany, and most other European countries. Very surprising to me. And that was one reason why I felt there was a need to influence a change in that attitude and make the statistical community in Europe a full partner and contributor to future European approach. So I created a bi-weekly 2-hour lecture series on statistical aspects of drug evaluation at the University of Basel's Department of Research. It was advertised in Switzerland, France, the UK, and Germany, and over 400 attendees came every other week. And the European course in pharmaceutical medicine continues as we speak. We created a course to continue the lecture series, and it's been going on for 30 years, and it's had well over 2,500 graduates. And this is the website for this particular European course in pharmaceutical medicine. This is the website for the European course in pharmaceutical medicine. If you want to go there and find a little more material on it, essentially these are approximately 2-year courses that have 6 modules that meet every 3 or 4 months. So you can read more about this by going to this website. Again, when I was at the FDA, I was living in Boston and telecommuting to the FDA home base campus in Silver Spring. But for 4 years, I taught a course at the Harvard School of Public Health called Quantitative Methods in the Evaluation of Evidence. And I co-taught this with Marcia Testa. And we wrote an article in Chance magazine that described the approach, the content, the case studies, and how to access and utilize content available on FDA's website. It was intended to help academics, professors find material that they could incorporate into their classwork. This is a screenshot of the article that we published on this course. We published it in Chance magazine. And it was based upon 4 years experience with a spring course at the Harvard School of Public Health. And the title of the article was "Let's Go to the Videotape, Promoting the Visibility and Understanding of Regulatory Science by Using Case Studies from Publicly Available FDA Information." So if you read this article, it's about 8 to 12 pages long, I believe. It provides a number of case studies and provides some information as to how to get access to the videotape that were the advisory committees that incorporated the case studies that I'm talking about. So the key takeaways from this course was that I had a multidisciplinary student, graduate, postgraduate population. They were statisticians, epidemiologists, in fact, several lawyers, some clinical investigators. But the problem I found was this course, even though it was a graduate course, it got access to students who were too early in their career to fully absorb the regulatory science material. My feeling was you probably had to be in the industry for 5 years or so to understand the depth of the material that was being taught. It was also a challenge to include all of the material in class versus out of class. So there are a lot of assignments that could be provided to students outside of the in-class work that could benefit from their doing it at their leisure. And one example of this is the COVID-19 Zoom experiences that you could go to that are on YouTube to get some idea of how you might understand the level of discussion for the evidence behind the vaccines for the COVID-19 vaccine. The FDA Advisory Committees are really powerful tools to illustrate decision-making and evidence evaluation. I think that there's no other opportunity to experience that the NIH application review process or the peer-reviewed literature process doesn't match what you would get exposed to in some of these FDA Advisory Committee videos, in my mind. So the regulatory science space contains a huge amount of material relevant to the pharmaceutical scientist, and it's dynamically changing over time. And I'm unaware of any academic curriculum that embraces any aspect of this. So this is not regulatory affairs. This is regulatory science, which essentially is about the science of it. So my experience with the European course in pharmaceutical medicine and the Harvard School of Public Health course, in my mind, provides a foundation for what new educational models might look like and how to launch them. Unfortunately, pharma had a training center at one time, about 20 years ago, and pharma reorganized themselves and I think lost their purpose and missed the opportunity of actually incorporating some of this kind of material in training their own pharma scientists. The biopharm section of the ASA is very active and actually incorporates some of this material in their programs at annual meetings and other venues. DIA webinars are very useful and are active, particularly the adaptive design webinars. The NIH CTSA network is a possibility, but they're difficult to engage in this particular topic. They actually have access to the 45 to 50 medical universities around the country and could be a vehicle for this, but I've tried to knock on their door with not a whole lot of success. And so departments of biostatistics, I don't know how this material could be incorporated in coordinated networks. It's more than any one department can deal with themselves. But if there was some shared coordinated networks, that might work. I don't have any more brilliant ideas as to how this could be accomplished, but I throw it out there for others to think about. So what's happening in Europe that actually recognize the need for new ways to bring the academic community into the regulatory science field? I have found it quite amazing that the European funding mechanism and has coordinated coming out of the European EMA, European Medicines Agency, has actually thought about this. And here is an example of that on the EMA's website. And it's under the area of innovations in medicine. And I was aware of the efforts that have been going on in funding by the European Medicines Agency for the last few years, but I was unaware of a recent initiative. And that initiative is exactly dealing with the topic that I've been talking about. And it's on the topic of strengthening the training of academia in regulatory science. And they have actually funded this. So the short term is STARS. That's for strengthening training of academia in regulatory science. This is the website that you can go to and find out more about the content and how they are implementing this idea. So I leave it to you to follow through on that. This is more of the same. It's a hard slide to read because it's small print, but it goes into where and how the project STARS has developed an online inventory to assist European academic drug developers in finding the various support services provided by this particular coordination effort. I find this quite amazing. I was unaware of it until more recently, but I think it's a model for how maybe the United States might be thinking about bringing academic communities into regulatory science. So training to complement discipline-specific education and to foster team collaboration is sort of my mantra here. Get beyond the checkbox approach in good clinical practice training. There are a lot of efforts that train the clinical trial team, but I'm not so sure they're being absorbed. The training is absorbed the way it should because it's very much a checkbox. Well, I've covered this training, check it off. So the concepts embedded in the ICH updated guidances E8 and E6, which are quality by design for clinical trials, are very much of a prospective culture change in actively identifying in advance the risks to clinical trials and planning to manage those risks to a study. And in my mind, this proper training would emphasize and force the team to stress test the assumptions of clinical trial designs, including scenario planning and simulations. And again, one of my points is that who would be the lead to do these stress test exercises? My position is that if you have experienced statisticians who have been around a while and seen many of these examples, my position is that experienced statisticians are the ones who are the most well-positioned to lead the team on stress testing the clinical trial planning evaluation effort. So again, just to summarize, at the conclusion of this talk, here is the publicly available regulatory information that forms the content of regulatory science that could be incorporated into a learning model. These are the links for the FDA website for guidance documents, the FDA website for documentation of review decisions, the website for publicly available webcasts. Here's the ICH website and the European Medicines Agency website. And finally, there's been, because of the COVID-19, many lessons learned on what the new initiatives are, collaborations, and recommendations to be ready for a new pandemic when, and hopefully we don't have to face it again. But this is a webinar that you can go to that talks about the lessons learned in the clinical evaluation of therapeutics for COVID-19. And it talks about the value to collaboration, moving quickly, changing the whole culture of how we approach collaborative research. So I think that as a result of this culture change and the need for and the value of collaboration to develop effective therapies to meet critical time-sensitive therapies, not just vaccines, but the COVID-19 example has actually moved the needle to talk about the value to collaboration among many stakeholders stimulated by a crisis. And I think that regulatory science is one of the things that could move into this space and maybe get a stimulus from the crisis that we faced with the COVID-19 experience. Much of my talk would have been about some new ideas, but the COVID-19 experience has just made them more relevant. And how do we respond in a timely way to evidence generation? So my concluding remarks are the following. There is an opportunity for new learning models and regulatory science content to be introduced to the scientific and clinical community who eventually enter pharmaceutical development and regulation. Moving the needle to get there will not be an easy task without a commitment. Statistical leadership is important and critical to that happening, and I believe the talent base is out there within the statistical community to do that. Experienced statisticians are good candidates to move the needle, lead teams, illustrate the value to prospective planning, simulations, and show how the content available in the public regulatory space fits together and is integrated. And with that, I think I am done. I would welcome some questions if you have them. Thank you.